Welcome. Good morning, good afternoon, good evening to our guests from the US. We've got people joining us from across Asia, Australia, New Zealand, Pacific Islands, the US, of course, and further afield, I'm sure. So please take the time to introduce yourself in the chat, particularly if you're not from one of those countries or regions. I'm very pleased to be the moderator today of this session. It's a really important discussion on fall armyworm and resistance. My name is Alison Watson, if you don't already know me, and I'm the head of the ASEAN Fall Armyworm Action Plan Secretariat. Before we start the session in full, and we have a formal welcome from our co-moderator today, Dr. Ramesh Puri from the Department of Primary Industries in Queensland, I'm just going to take the time to go through and tell you how we're going to use the Zoom platform today. So just to note that the session will be recorded. We will have a copy of the speaker presentations. We usually share them around 1 week after the session. And we have a copy of the videos and we load them on a YouTube site, and you'll be able to catch up with the videos there. If you have any problems today, the best actual thing you can do is probably to log off and log on again. So there's no sort of flash technical advice here apart from doing that. It usually works well. You can also go to the arrow where the audio button there is and have a play around with your audio settings if you're having some issues there. You can also send us a message to the chat box, but we may not be able to help you too much with technical, but we'll do our best. And that is about it, apart from watch out for that shared video and PDF of the presentations in about a week to your email box. So the way we are going to be active today is really through the Q&A box. So if you have questions, and we hope you have many questions because we have these experts lined up for you, grill them, ask them anything you would like around resistance and fall armyworm, and let's see what they can help us with. They have many years of experience in the field and in the lab. So it's brilliant that they're here, but please just make it easy for us to answer all your questions by putting them in the Q&A box. On the other hand, we do really like to have a big conversation and we like to know what you're working on, and we also are keen to see anything that you can share with us, latest papers, reports, a project that you're working on. If you could put that in the chat box, that would be great. And don't hesitate to give some reactions, clapping hands, a heart if you really like a particular presentation. Feel free to use that function as well. I'm just going to do a quick poll. It's really just to sort of get to know you. Let me see if I can launch it. There is 2 questions here. And hopefully you can see that. There's 2 questions. It's anonymous. We're not recording. Hopefully that's come up. Can I see anyone answering it? Yes, I can. That's fantastic. So the first question is just, have you seen fall armyworm within the field recently, within the last 2 months? And if you're one of our speakers, I don't think, unfortunately, you can answer the poll. But if you're one of our guests out there, 177 at the moment, please take time because it's interesting to know who is out there seeing fall armyworm right now from across the world, probably. And the second one is related to the topic of today. So how confident are you that fall armyworm resistance is well managed in your country? And let's see what people have got to say, and that's what we'll be talking about further. So I'm going to give everyone another 20 seconds. We've got actually over 100 people already answered that short survey, so that's fantastic. I'd love to get up to 70%, so if there's a few more people there. I'm going to end the poll now. And I'm going to share the results just so that our speakers can see as well. So have you seen fall armyworm within the field recently, within the last 2 months? We have, yes, 55%. So it would be great to know from that 55% who answered, in which country are you seeing fall armyworm right now? I know that in New Zealand, the season is just starting for fall armyworm, so there might be a few New Zealanders there. It would be great to hear from the Australians and also anyone in Asia around what you're seeing. And two, how confident are you that fall armyworm resistance is well managed in your country? We have quite mixed results there. Extremely confident 4%, confident 24%, not very confident is the bulk of people, 45%, not confident at all, 10%, and not sure, 18%. So mixed results, but obviously a little bit to go there to get to full confidence. So it's a good start today, and we'll be asking some of our speakers more about what other things we can put in place and what are the lessons learned to better manage resistance in fall armyworm. So thank you very much for answering that. What I'm going to do now is I'm actually going to just tell you that it's a very fast-paced session again, and we will move through quite fast. So please put your questions in that Q&A. Here is the agenda. We're going to start with David, and then we're going to move to Dominic, and then Andy's going to end the session. 3 international experts in this field, and it's really pleasing to see them join us, and I really appreciate your efforts in the evening to join us, and also in the morning, Andy, to join us from Indonesia as well. So what I'm going to do now is I'm going to actually welcome Dr. Ramesh Puri to give us our formal introduction to the session. Ramesh.
Thanks, Alison. Excited to have this number of participants into this webinar. Hello, everyone. I would like to respectfully acknowledge Aboriginal people and Torres Strait Islander people as the traditional custodians of the country on which we meet, and I pay my respects to their elders past and present. I extend that respect to all Aboriginal people and Torres Strait Islander people here today. Thank you for joining today's webinar on fall armyworm and resistance management. I am Ramesh Puri, Extension Officer with the Department of Primary Industries, Queensland. I am working on the National Fall Armyworm Innovation System for the Australian Vegetable Industry Project, which is funded by Hot Innovation using the Vegetable Research and Development Levy and funds from the Australian government, with co-funding from the Queensland Department of Primary Industries, Victorian Department of Energy, Environment and Climate Action, and New South Wales DPAD. As part of this project, insecticide resistance baseline monitoring for horticulture and the development of a resistance management strategy are currently underway. The overall project purpose is to develop and sustain a nationally coordinated fall armyworm area-wide management innovation system for the Australian industry, leading to increased capacity for sustainable fall armyworm management beyond the life of the project and capacity to manage future threats. It is a privilege to welcome participants from across the region and to join with Asian Fall Armyworm Action Plan, Dr. Ellison, in supporting a shared effort to strengthen resistance management and protect the effectiveness of the fall armyworm management tools. We are fortunate to have an excellent panel of speakers. Dr. David will open with insights on staying ahead of resistance, followed by Dr. Dominic with field-level perspectives from grower systems, and Professor Andy with insights on risk regulation and the responsible use of management options for fall armyworm. So your experience and knowledge will provide a strong foundation for the practical discussion that will follow after the webinar of each speaker sessions. Thank you for being part of the session, and welcome again to the webinar. I'll now hand back to Alison to guide us through the agenda. Thank you.
Thanks, Ramesh. And also just a big hello to Dr. Witek Tay as well, who's joining us in the background there to help with technical questions as well, who's been a big supporter of the Fall Armyworm Action Plan in ASEAN. And just a quick note on that, it's turning into the Bio-Innovation and IPM Action Plan, ASEAN, as well. And there's some links there that I will share later with links from also the Australian work. But a big theme of this new action plan, or phase 2 of the ASEAN Fall Armyworm Action Plan, is going to be regional resistance. So resistance management is a big theme, and it's really around managing resistance at local, local, country, and regional level, and actually making sure that they all support each other, that resistance management plans and actions and activities. And so that's something to think about as we're guided through the sessions today from the speakers around how important it is to really manage resistance in a coordinated and collaborative fashion as well across different landscapes and at different levels. So just to update you on that, and that really brings me to our first speaker. So David, we're very excited to have you join us. I'm going to get you to load your presentation. I'm going to stop share. And David is joining us from University of Michigan. So it's very cold over there. We were just talking about how it's quite a lot in the negatives. I think it's -10, maybe -20 at the moment. So it's cold, but you don't look cold there with that beautiful monarch butterfly behind you. So welcome, David.
Thank you, Alison. Thank you, Ramesh. Thank you for the invitation. It's an honor for me to present. So I'm going to speak about my experience working with Fall Armyworm. And the title is Staying Ahead of the Resistance, because when I see the poll that Alison conducted, it's like the optimism is not very good. So hopefully at the end of my talk, it's like looking at the future in 5 to 10 years in many, hopefully not in many places of Asia. So this is the outline of my presentation. I'm going to give a little bit of brief background on resistance globally. And then I'm going to talk about especially Puerto Rico, fall armyworm in island type conditions, and resistance monitoring in Puerto Rico. So I don't need to repeat a lot of all of this because you already know this species, the origin of this species from the Americas. The Mayans, 12,000 years ago, this is an indigenous people in Mexico, managed the fall armyworm taking the larvae from the plants. You know, the core is the origin of the core is from Mexico. So, but this pest distributed all over the world, and then it's a big problem. This is crops treated with pesticides and with pyrethroids and other treatments, and you can see the terrible damage caused by fall armyworm. And here in the middle, you see some plots treated with, at that time, emamectin sulfate. Now this compound failed in Puerto Rico. And then now in our pesticide resistant database, fall armyworm is placed in the number 18 as is inside of the top 20 more resistant species in the world. And you know, resistant, this is one definition of resistant. So I should have put a larvae, but basically there are few individuals that are selected after sprays, and after many sprays, then these resistant individuals start being the majority or the pest in the field. So, and this is a nice definition by Tabashne et al. So we have lab selected resistant, fillable resistance. So in some cases you can spray in the field and then you don't notice anything, but the resistance is present, like reducing the time of protection. And then we have practical resistance when there is a field failure. You spray and there is no control. So I'm not going to stop a lot in detail, but this is the status of resistance since 1914 to now. 637 species, almost 20,000 cases to 367 compounds from 1914 to now. So it's a big problem, this situation, and it's increasing the number of cases, like you see here in blue. So Fall armyworm has developed, there are 272 resistant cases reported in our database to 47 active ingredients and 8 monoactions. So there are so many different monoactions against Lepidoptera, and you can here see to the right the compounds, the monoaction compounds from acetylcholinesterase inhibitors to ryanodine receptor modulators. So this is the situation with resistance. And then if we see the historical perspective, since 1965, The first case of resistance was to Linden and DDT, and then continue to cyclodien, OPs, carbamase, pyrethroids, BTs, and then all the new compounds continue developing resistance. And the progression is going to be increasing the resistance because this is a problem everywhere, and everywhere is selected the Fall Armyworm. So this is a general view about resistance. So now I'm going to talk about a little bit the work that I do. In Puerto Rico is one of the most important nurseries in the world. This is a place for seed for the seed industry, for seed augmentation and research. However, in this place, the pest has the condition is that they have more than 12 generations per year. There is an isolated population, is an island. There is year-round core seed production, like a 3-4 seasons, and then one of the things that happen in this area is like there are 30 sprays per season. So it's about 90 sprays per season. So imagine the high selection pressure that is occurring. And it's one of the places that occurred the first case of field evolved resistance to Cry1F in core in 2003. And then in 2008, they stopped planting core using Cry1F to control the pest because the problem, but they need to use other pesticides. And from then, there is many situations. So I have a group of talented students that were in my lab. I'm going to talk a little bit about their work in Fall Armyworm. But for example, Rebecca compared populations from Mexico and Puerto Rico, comparing two different systems with low sprays of pesticides and a lot of migration in Mexico, and then with relative isolation in Puerto Rico with 29 sprays per season. So the methods that she used was topical application in the case of insecticides, and then for some assays to BTs was overlay dye assays to these BT toxins. And then here is some of the results. Resistant to Spinoteran, Chlorpyrifos, Metomil, and Permethrin. I never see, and then you can see the rest of the susceptible population. This is the resistant ratios. But you see in other places of Mexico, low levels of resistance, but in Puerto Rico, really high levels of resistance. You see Metomil, and then high levels of resistance, OP, pyrethroids. And then this is a situation that is really, really complicated. And then when we see the diamide compounds, there is extremely levels of resistance to fluindamide and chloranantraniliprol. And then you see other samples from different geographical regions in Mexico and very low levels of resistance. So another way to see this resistance is like when we have something that is called relative reduced potency. If we, for example, in this case, the susceptible colony, we need the LD50, we need 5,000 times emamectin benzoate to have the LD50 of amectin. Abamectin is not a very powerful compound. But in the case of resistance in Puerto Rico, the thing that happens is that you need less compound that reduce the potency of the compounds. I'm not going to start a lot, but you can see all the compounds, different types of monoaction compounds. But here you can see still emamectin at that time was very effective. And then to the BTs, I mentioned 2003 was introduced the Cry1F in Puerto Rico, and then Nick Torres find resistance. This is a population sampling in 2017, and you see almost 10 years later after stopping the planting or core with Cry1F, still the population at 10,000 nanograms, there is less than 3% of mortality in the resistant population with Puerto Rico, and here is the susceptible. We tested other BT toxins, and they were resistant to Cry1AC and not resistant to other ones like cry2A, other type of protein. This is the situation. We continue these studies with similar system. We dye overlay dye assay. Omar Pozos performed assays, found resistant to these compounds. But the interesting thing to do is, well, what happened if we cross the resistant with the susceptible? What is the situation? And then we found that it's codominant the inheritance of resistant flubendanide, chloranantraniliprol is similar situation. So what happened with this? In many places of Asia and many places of the Americas, the mice are used to treat the seeds. So the pest needs only one copy of the gene. When the residues decay, the one copy of the gene is enough can be enough to protect the larvae, and then killing is removing susceptible the susceptible alleles, and then you are going to find fast resistance. So this is a valuable impacts for that. We go further in our research, and we use different inhibitors, so different enzymes, and this is resistance to I think that this is SM Valere, yeah. And then here is the susceptible population in your left, and we use different inhibitors, so different enzymes. And your right, you see this inhibitor suppress P4 Fichter, glutathione transferase, esterases, ABC transporters. So meaning that this has a high potency. The insect would utilize the metabolism to detoxify pesticides. And then because the origin of fall armyworm in Africa, in India, and many places in Asia came from the Americas, so it's very likely that some of these genes are present in the other side of the globe. Other system that we are working is with botanical compounds. We are working with host plant resistant, with mycin, but also we are working with neem, with azadirachtin. And we found resistance to azadirachtin, to neem. So this is the first case, a field ball resistant or any species to azadirachtin, the main compound, neem. So it's quite interesting, this situation with the fall armyworm. But remember, this is a pest that is exposed to high artificial selection pressure. So what happened? Well, we had all of this, and this is important. If I were in Australia and other places, I compare using the same method and compare the data of that populations with my data with the susceptible and resistant insects. So we perform a training. What do we do? We perform training in Puerto Rico. IRAC and Pravia, this the organization for seed. We are collaborators. We perform pesticide applications in the field. You can see the plots and the damage. This was emamectin. Unfortunately now, emamectin, my PhD student Shatrughan Shiva from India found 30,000-fold resistant to emamectin sulfate. So it's very high levels of resistance. But we perform different assays, and we work with the industry and training them how to perform bioassays, and many of the industry now is performing assays. So there are many recommendations from this workshop, like it's important to target the early instar, it's important to reach the word of the plant, the application in the word, the amount of water, the adjuvant. And then it's not like the industry doesn't do anything. There are sampling, there are a threshold to spray, there are release of natural enemies. And one important thing that has been really helpful to manage the resistant larvae is the use of nematodes to control the resistant larvae. So the nematodes can restitute or at least can be really helpful to manage the fall armyworm larvae. So in conclusion, this is something, the data that I have more data. I have a lot of data with so many different pesticides. And the resistance levels this is field evolved resistant. It's not a population that was selected in the lab. And most of the cases that we have is resulting in practical resistance. And then if you see this data, this situation might happen, hopefully no, 3, 5, 10 years in many places in Asia because the similar conditions, tropical weather, island time conditions, many pesticides these specialty compounds are very accessible to many farmers. So that's might be a problem. My prediction, I know somebody told me that I'm not very optimistic, but I think that the Fall Armyworm is going to move to the top 10 more resistant species to pesticides in the world. And then our data, feel free to use our data to compare your levels of resistance. We have susceptible colonies and we have resistant colonies. And I would like to mention that rotation insecticide is really important in an area-wide management. And then, of course, it requires more tools, no? From the IPN tools require more. In this case, the nematodes was really important to control the larvae. Of course, that this is not possible in some cases because it might be costly to apply nematodes. But I consider that this program benefits Puerto Rico and other places of the world that can compare this accelerated resistance with the resistant levels. And with that, many thanks to the organizer and thank you. And I have so many different people to acknowledge, people from the industry, from the researchers, colleagues, Bruce Tabajny from University of Arizona, and other collaborators around the world. And then I think that I had 15 minutes, so if there are questions, I would like to entertain any question.
David, that's perfect timing. Thank you.
Uh-huh.
Yeah, you're a fantastic speaker.
Thank
Right on
you.
time. No, really interesting. So this is correct if I heard rightly. 29 sprays per crop, per season. That seems an incredibly high amount. Why is that? I mean, is there a behavioral aspect there that you also looked at with farmers, just sort of seeming to spray a lot? Were they not looking at the action threshold? Were they just doing a weekly
No.
spray? Or why was it so high?
Well, the thing that happened is that this place is special. You see the crop, there are so many egg masses. There are so many egg masses. There is an intense selection pressure. And then just remember that in Puerto Rico, it's not like a place in the Midwest or in many places of the US. The core is for core production. This is a place for seed augmentation. And we're talking about 6,000, 10,000 acres, or sometimes less. So it's a small area, but every plant has incredible value. But regardless of that, even if it's for core production, that there are more wholesome, many things is the selection pressure is very high. I remember I went to Myanmar in the first wave or attack of the Fall Armyworm. I found many plants with many egg masses and a lot of pest infestation. So in this case also, the pest pressure is very high. And then there are thresholds. Of course, that there are thresholds, and the people use many monitoring. They use traps. They release natural enemies and all the things. But just remember that to have a nursery and to have the facilities is very expensive. And it's not like I'm going to say, okay, I'm going to do not to produce core in 2 seasons. It's not possible that because there are so many investment. Or for example, I had experience from Mexico. In Mexico, there is the cold in the winter. There are no crops and there are no plants, and it's a mild, really mild. They call winter. It's a really mild winter, but there are no pests. So that reduces the development of resistance. But when you have a pest that is present the whole time, and then you have other vegetables, and other people spraying, then you have a continuous pest pressure.
Actually, that's another question I had as well, is what you talked about it on maize, obviously, for the session, but what other plants or crops are you finding fall armyworm on?
If I remember
Puerto
well,
Rico.
tomatoes, And there are other grasses also that can be a host. You know, fall armyworm has more than 300 hosts. So and then it's tropical weather. The temperature is perfect for them. So there are seasons that is more pest pressure, but there are other seasons that is lower. But in general, you can find the pest the whole time.
Okay, excellent. I'm going to throw it over to Ramesh. Ramesh, do you have a question?
Thanks, David, for this presentation. Mentioning about the area-wide management, rotational mode of actions, which is playing an important role on that management perspective for the area-wide aspect. So given your global experiences tracking insecticide resistance trends, what early warning indicators should be prioritized to stay ahead of resistance development in fall armyworm?
Well, of course, it's important to have a monitoring program, and you compare the data with the susceptible. And then there is this is really interesting to have a monitoring system. But in the field, when you start, even that the compounds control the pests, you start seeing damage in the leaves. So this is an indication that the compound is in process or resistant. For example, I see emamectin benzoate in 2020 or 2019, and the crop was perfect, clean, all clean, clean, clean. And then we saw indoxacar, we saw diamides, and we start seeing all the damage. Pyrethros was the same that the control. So in my opinion, there are some indication that there is a reduced control, and that might be indication of early warning or resistance. But when that happens, is you will notice in the bioassay that the resistance levels are high.
Thank you.
Okay. I've got another question here from the audience. What mode of deployment do you implement in Puerto Rico when planting BT corn? Bag in a bag or mixed seed blend?
Could you repeat, please?
What mode of deployment do you implement in Puerto Rico when planting BT corn? So there's a few questions here around refuge and also whether you're...
I don't remember well the part of the refuge at that time when was deployed the Cry 1F. Um, because there are so many hosts outside, so I don't remember exactly, but the situation is that the resistance developed very fast. I don't think that it was a very effective refugees, but I don't remember exactly, but I can send back the question. I can look into that, and I can send the answer to.
Okay.
But my impression is that there is no an effective refuge and that happening, and then continue. We carry 1F in many cases or resist. Dominic can speak more about this because he's the expert. But in many isolating cases, I continue planting or carry 1F, there is a development of resistance, fast development of resistance.
Okay. And one more question here. There's quite a few questions here that interest in nematodes, and I think you suggested that you were using in the nematodes, or they were using that in Puerto Rico, and they were quite successful. Can you just give us a little bit more sort of
Well, it's
information?
Sternonema carpocapsa, the compounds produced by copper, yeah, copper, and then it's using mixed with insecticides. And actually, I have a PhD student now working in to study the mode of action, and then if it's synergism, antagonism, or additive effects in the resistant and susceptible, because there are insecticides that actually some insecticides or relating insecticides are used to treat the nematodes that attacks mammals. No? So we're looking at different aspects of that, but we know that the nematodes by itself is not so effective. But when it's missing insecticide, made a mediocre insecticide to restitute part of the control.
Excellent. Thank you. Thank you very much, David. And we're going to end there. We're going to come back hopefully with a question at the end. You have quite a few questions in the Q&A, and if you could go into the Q&A and answer any of those that you would like to, that would be most appreciated. Thank you so much for your presentation. Very interesting to see that case study as well in Puerto Rico. It's just fascinating just how much pesticides have been used there, and not surprisingly, just how quick the resistance has developed. So it's a good segue into introducing our next speaker. So if you could stop sharing your screen, David.
Yeah.
And Dominic, you are next up, and you are an expert in this resistance in BT, and you've got a very interesting presentation today that I've seen some of not so long ago in Malaysia that really talks about working with farmers as well. So I know that's going to be quite an interesting point for many of our listeners in the room. So welcome.
Hi, thank you for the introduction. Yeah, that's right. My area of expertise, at least in resistance, is on BT. And in the United States, we manage fall armyworm and other lepidopteran pests in maize and cotton with BT. And so that's kind of going to be the focus of my presentation today. David did such a great job of defining resistance, but one of the things I want to frame it, because I do a lot of work with growers themselves, is to ask the question about, what is so bad about resistance? Obviously, there's economic consequences. This photo here is actually taken from a cornfield that was infested with fall armyworm late in the season, and you can see the ears that actually fell off the plant in the middle of the rows there. There's environmental consequences because growers often bear the economic consequences of resistance, but the environmental response is usually to spray more foliar insecticide, and then sometimes we have cross resistance between different modes of action. So it's very important to delay resistance using different methods, but there's multiple methods that we can think of. The first being the regulatory structure. What incentives are there for growers to delay resistance? What methods of enforcement are in place, or what are they lacking in the structure? Obviously, the seed companies have a role to play. Are they invested in delaying resistance to their products, or do they have to compete with competitors in the marketplace? Who are the players out there? Grower behavior is something I'm going to focus on later on in this presentation, but of course, that's very important in resistance management. There's supply chain realities, and that, let's say growers want to do things like plant lots of refuge crops. Are there good varieties available for those growers? Are they easy to obtain? Do they yield well? Who do growers obtain their advice from? Is it from independent crop consultants? Is it from crop consultants from the companies? Is it from people that they sell the seed from? Is it from the government? All these things can influence resistance. The pest ecology is very important. Whether a pest disperses locally within the field, whether it migrates across the landscape, what sort of host it uses. These are all very important in resistance management. Landscape composition, are those hosts close together? Are they spread apart? Are they present together in time and space? And then how coordinated is resistance management on the local level with the growers themselves, on the regional level, and then even on the international level, which is something that perhaps you have to think about in Southeastern Asia as country sizes change. You have barriers of mountains or waterways or things like that. And this can all create a big headache when we think about resistance management. Which of these is more important? And that really the answer is, of course, it depends. So I want to start this presentation by just giving 3 really brief examples of resistance management to set the stage. The first example I want to give is one of strong resistance management. And this is an example from the Australian cotton industry. They have a very important pest there. There's 2 species of Helicoverpa. There's Helicoverpa armigera and Helicoverpa punctigera, with armigera being the most important for cotton production. And this almost took down their cotton industry in the 1990s because of resistance to foliar insecticides. And so the growers there realized that when BT cotton came in, they were going to have to do a really good job of stewarding that product. And you can see the areas of cotton production in Australia on the map here. One of the things that made their resistance management so successful was mandatory enforced stewardship. So this was things that growers were required to do in terms of what BT crops were going to be used, in this case, cotton and not maize or corn. They also had an area-wide management strategy where growers realized that they not only needed to cooperate in a single cotton producing areas, but other cotton producing areas needed to cooperate as well. They understood the insect ecology and the landscape very well. They knew what hosts it used, they knew how it migrated. And I won't go into detail, but when they didn't understand the host ecology, they did studies on it to understand how they could improve resistance management. And I think very important for this story is that they had industry-wide cooperation, but it was really the growers were really behind it. The growers were the ones that say, hey, we really want to steward BT cotton and we want to see it successful. And they were then able to partner with their grower groups, the seed company, and the government as well. BT was really an advantage for them because they were spraying less foliar insecticide on the cotton, and so it helped increase biological control, which had knock-on effects for other pests. And they had very good monitoring systems in place where they were able to, as David mentioned, identify maybe those small genetic changes that were being taken for resistance over time rather than seeing field failures. So this was just a great example of strong resistance management. And I want to contrast that with a case that's room for improvement, and that's the use of BT corn or maize for fall armyworm management in Brazil. This is a very challenging pest to manage because most parts of Brazil are tropical, and so that means that this pest has multiple generations per season. There's often multiple crops in a given field in the season. And so there's this sequence of crops that the fall armyworm can use and just be really, really exposed to these BT toxins over time. It's almost the exact opposite of the Australian case where rather than mandatory stewardship, there was no enforced stewardship. There's no really cooperation between growers, either on the area or within the industry. I've already mentioned what a challenging place it is to manage a pest like fall armyworm. Insecticides are used very frequently and kind of complicate the system. And unlike the Australian example where there was a good system of monitoring in place, in Brazil, fall armyworm BT resistance was only discovered with field failures. The last example of thumbnail sketch I want to give is from the United States. And the United States, I would say, is probably somewhere between the strong management, the strong stewardship example from Australia, and the major room for improvement from the Brazil example. And the example I want to give is from a pest that's related to Helicoverpa armigera, that's known as Helicoverpa zea. And it's a pest of cotton, corn, soybeans, sorghum, a number of for different crops. But importantly, in the US, both BT corn and BT cotton are grown in the same areas. And this pest uses both of these crops for a host, and both of these crops express BT in the United States. So that creates a sequence of hosts that the insect can use to be exposed to BT traits multiple times over the course of a single season. In the United States, stewardship was very good for this pest early on, but it soon eroded over time. Just changes happened in the system where it wasn't quite as good as it once was. There was no area-wide management between growers or across the industry. I've already mentioned how challenging it is of a landscape because you have this sequence of hosts that express BT over time. Industry did some cooperation, but in the US, they have to register these traits individually with the United States Environmental Protection Agency. And because of that, there's individual resistance management plans for each trait rather than a single overarching one across the system, across companies. Another problem was that there were single BT products that were introduced and then pyramided ones that were introduced alongside of them, but there were overlapping times where these single and pyramided products were planted, meaning that the insect was exposed to these single traits over time that eroded some of the traits in the pyramided products. And then resistance was discovered with monitoring early on. Nothing was really done about it, and we weren't able to react until there were field failures that were present. So this kind of might leave you sitting there with your wanting to tear your head out. We've talked about how complicated it is to manage resistance, and we've given some good examples and bad examples. And in the US, we have a fairly unique extension system compared to many other parts of the world. We have people that work on the local level in extension, but we also have people in the extension system that are attached to the university. And I'm one of those extension people that works in the university. And really, one of the very few things I can actually do about resistance is to change grower behavior. And regarding Bt resistance, one of the easiest ways I think to change grower behavior would be to get them to plant more non-Bt refuge. So in the area of the United States where I live, growers are required to plant to their total corn acres 20% to non-Bt corn hybrids. And so to change grower behavior, it's important to know what growers care about. Of course, growers care about yield.
5 minutes, Dominic.
Thank you. But they also care about profitability and sustainability. So if we can take advantage of these things, we might be able to encourage growers to delay resistance. So one of the ways that we've been doing that in the US is to help them evaluate the yield of non-Bt hybrids relative to Bt hybrids. This is an example of a small plot trial that I conducted on a research station. I've conducted this same trial for maybe 7 or 8 years. We've done this in what's called the official variety trials, where the university plants small plots in different things across the US. We've combined data looking at the yield increase of Bt corn relative to non-Bt corn over time, and we've seen some effects by planting date. And another thing we've done is that we've actually taken these hybrids and put them on the grower's farm using the grower's equipment. So we've said, hey, here's a bag of Bt seed, here's a bag of non-Bt seed, plant this on your farm with your equipment, take the yield on it, give it to us, and we'll look at the profitability. And when we do that, we often see that the profitability of planting your farm to 100% Bt versus the required refuge, 80% non-Bt and 20% non-Bt over time, is the same. The final thing I want to talk about today is grower education, because it has often been suggested that we just need to educate growers more. If we give growers more information on non-Bt, we can get them to change over time. So I've actually done this. I've educated growers, and I surveyed them before and after I've educated them. And I found that only around 40% of growers plant non-Bt refuge, and that I'm able to change their knowledge in the short term. They understand refuge important, they know more about it. But in the longer, medium term, I was not able to change their willingness to plant refuge. And quite disturbing is the smaller growers are actually less likely to plant refuge, and there's more smaller growers out there. So it seems like this kind of big intractable problem. So one of the things we've done is we've reached across the aisle to social scientists, and we've said, how can we change grower behavior in this knowledge deficit model? And one of the things we tried was an emotional appeal. This emotional appeal is very simple. We simply remind growers of the importance of cooperation. We give them little stories, little vignettes about how cooperation is so important. And I'll skip ahead to my collaborators here. This is Zach Brown, who's in the Bio and Ag Resource and Economics Department, and a very talented PhD student of his, Greg Ferraro. And one of the things that we did recently is we measured conditional cooperation in Eastern North Carolina. Conditional cooperation is simply the idea of, I will cooperate, but only if you cooperate. We did this in areas that are poorly drained because we wanted to see if we could layer those two things together. Perhaps in areas where growers are already cooperating in drainage, they may be more willing to cooperate to plant non-Bt refuge corn. And we
One
did this
minute,
through, okay,
Dominic.
we did this through our extension efforts. Very simply, we had them play a game. The growers, we measured conditional cooperation in this game. Basically, we were able to tell which growers were willing to cooperate and which were not. And then we layered our emotional appeal on this. Half the growers we gave the emotional appeal to, half we did not. And I'll skip to the chase and let you know that the emotional appeal worked, but only in areas where growers were not planting refuge. The growers that were already planting refuge and cooperating were less responsive to our emotional appeal. So, in conclusion, we want to think about what might serve as proxies for cooperation in your areas. And what I want to encourage you to do is to look for areas where growers are already cooperating. These are areas where they might be more willing to plant non-BT refuge. And just remember overall that biology sets the rules, but behavior decides the outcome. That is to say, no matter how good your plan for refuge is, if the people that are behaving in the refuge are not cooperating or doing things together, it won't work. Thank you for your attention.
Dominic, thanks. That's excellent. Really interesting at the end too. It's fascinating. Your work with farmers there and just how important understanding behavior and understanding how to communicate and communicating in different ways can actually help influence decision making. In some Southeast Asian countries, in 3 countries, there is BT maize. In Australia, New Zealand, and other countries, there is not BT maize yet. But I'm sure what you're sort of looking at here around how to appeal to farmers, how to communicate, how to make that relevant would apply in those situations as well. So for around, changing mode of action, reducing unnecessary pesticide use. Have you seen any sort of more, or have you looked at that as well, or any thoughts on sort of in that sort of situation, how you might develop sort of a behavioral sort of approach or appeal?
No, we haven't expanded this work to other systems, but I would like to just say that the area where we measured this, Eastern North Carolina, is a pretty challenging area to measure behavior because these growers they're not very cooperative, right? And some of those areas you mentioned, I imagine, like for example, Australia, where they're already used to cooperating across the cotton industry, this would be an easy sell. But in other areas where they're not used to cooperating, I think you'd really have to kind of dig in and figure out how could we craft a message around the importance of cooperating to make this thing work, and here's why it's so important to work. But we haven't expanded it beyond just what we've done.
Excellent, thanks. There's quite a few questions coming in. I'm gonna throw it over to Ramesh. While I'm throwing it over to Ramesh, just to make sure that we're using our time effectively, Dominic, can you just stop share with your screen and I'll get Andy, our next speaker, to load his up. Ramesh, have you got a question for Dominic?
Thanks, Dominic. As an extension officer, it just reflects to me what you have been doing about the behavior and about the knowledge part of this fall armyworm management. We have an area-wide management and we are working with the industry on area-wide management perspective. And with the new variation into the project on insecticide resistance management, it is totally reflecting what we have been doing, applied in our case, but we are in the horticulture sector. So from your work with growers, which practical field level decisions have the greatest impact on slowing resistance development while still maintaining effective fall armyworm control? Can you please share it on that?
Yeah, that's a great question. And I should say that for growers in North Carolina, it would be planting non-BT maize. But we have a huge advantage in that when we plant non-BT maize, we don't have extremely yield limiting pests that come in when we plant it on time. If I were to give this presentation in Brazil or I don't know, Vietnam or something, right, where you would have a tropical climate, it would be a more difficult sell, I would think, because you would have to measure that with loss of the crop. So there's going to have to be some foliar or insecticide spraying that's layered into that message to go along with it to prevent massive loss to the refuge crop. But I think if you can identify the importance of the refuge, that'll go a long way to helping. The other thing I'll say too is I wasn't able to mention something called natural refuge, and David alluded to the fact that this pest uses 300 different hosts. If you can identify hosts in the environment that are non-crop hosts that are maybe contributing to susceptibility, that would be helpful too. So I think understanding the biology of the insect in the respective countries is going to be an important part of resistance management.
Excellent.
That's
Thanks.
correct. Thank you.
Thanks, Dominic. And we're going to come back with some questions, hopefully. What I'm going to do is I'm going to just quickly move to Andy, so we've got time to have his presentation as well, which is going to be really interesting because we're going to move right through to Indonesia now and see what's happening there. And then we'll come back with some questions. So just keep that thought. But thank you so much for presenting and really interesting, and it's fantastic to hear that work with farmers on the ground as well. So thanks, Dominic. Okay, Andy, nice to have you join us again. You've joined us on a few of these sessions, but it's going to be really interesting to have an update on what's happening in your part of the world. And also I know, for example, that BT maize there has been introduced now and is starting to be used by farmers. So that's an interesting development in Indonesia as well. But you're doing quite a lot of work in this space. So please, you're welcome to start.
Okay, thank you very much, Alison, and thank you for the organizing committee for inviting us to participate in this webinar. So I prepared this material with Ratna Rupiana. She is a doctoral student under my supervision, working on the Fall Armyworm. So let me start with the I'm sorry, I'm having a problem with moving my
That's
slide.
okay. Do you want to try loading it, reloading it again and see if that helps?
Still not working.
You can stop share, Andy. Stop the share and then we'll have another go. If not, I've got your presentation on.
Yeah, let me upload one more time and then
Yeah,
see.
yeah, let's try that.
Here you go. It's good. So just to give you an overview about the problem of resistance in Indonesia. If you look at the data and also the last publication made by the Andika and Martoro, they reported that only 3 insect species in Indonesia that has been reported resistant to insecticide so far. I think the way they collected the data based on the publication in the primary journals. We can discuss this in different aspects, but I do not want to get into the detail on this, but at least it shows to us that a very small number of report cases of resistance in Indonesia can be due to because of the research in resistance is not as intensive in other parts of the country. That's at least one of the hypotheses that I have. But the question is that, is there any resistance to Fall Armyworm? So I'm going to go through with 3 different aspects. First, we will be sharing about what is the most common practices for controlling the Fall Armyworm to give us the background. And then is there any resistance happening in the field and where to go from now. So this is what we did in 2021 during the COVID time. We did a survey funded by the FAW to look at on the Fall Armyworm management practices. And what we found at the time is that almost 80% of the corn farmers practicing monoculture, and insecticide was the main control measure, with the application of ranging between 2.5 to 5 times per season. And imamectin benzoate was the most dominant insecticide applied for Fall Armyworm, starting in 2019 when the first outbreak of Fall Armyworm happened in Indonesia. So looking at the pesticides that were recommended by the government at the time, there were 4 active ingredients, and then we look at on the 3 out of 4 just to see and to provide and to develop the baseline studies whether field population of Fall Armyworm, especially in Central Java, which is one of the corn center producing provinces in the country. I believe it's the second biggest. We're still susceptible to the 3 insecticides that we selected out of 4, which is imamectin benzoate, chlorantraniliprol, and spinosaurum. And luckily that we had a laboratory population that we were collected in 2019, which is the first time the outbreak happened, and we collected from the area where the corn is not or it was not very intensive in terms of receiving any selection pressure from insecticide. So we hope that those larvae that were as a founding population for susceptible population were really susceptible. So in 2021, we collected 5 populations from Central Java and then doing some bioassays using the diet tipping against the insecticide. And what we found is that all 5 populations that were collected in 2021 were still susceptible to imamectin benzoate, chlorantraniliprol, and spinosaurum. So this one is about 2 years after the first outbreak of Fall Armyworm in Indonesia. So what happened in 2025? So this is the part of the doctoral research from Rubiana. And she collected a population from Jambi. Jambi is one of the provinces and where she works at the time. And she collected 11 population, each one population for each regency. And some population have shown develop very highly resistant to imamectin benzoate. So I think if we connect with the previous slide showing that imamectin benzoate was the most dominant insecticide, and this was about 4 years after the use, and just probably this is the first data showing the field resistance of Fall Armyworm to imamectin benzoate. So the detailed data hopefully will be coming soon, sometime in the next few months, because the paper has been submitted and now is under revision. So at least I give you the information, the latest information that yes, resistance is happening in Indonesia to imamectin benzoate. What about BT corn? So as Alison mentioned, the BT has been planted by the farmers since 2023 and was intended for Asian corn borer, and with only one toxin, which is Cry1AP. And we all know that this is not the toxin that are very effective for controlling the Fall Armyworm. But I think the question is that, is the Fall Armyworm going to be resistant to this particular protein? Because based on our laboratory observation and also field observation, we very often found some larvae were still surviving on the on this particular BT corn. And I guess that shows us that the selection pressure is happening in the field. We just did not know yet at this time because we have not done any bioassay or other studies to look at on whether the population of Fall Armyworm that we have right now has developed resistance or not. However, on the other hand, we have done some baseline study for other BT protein that are used for Asian corn borer, and there are 4: Cry1A105, Cry2POP2, Cry1AP, and Cry1F. But as we might know that these 2 species is very often happen in the same field, probably in different plants, but different in the same field. So, and Fall Armyworm remains the one among 2 major pests in corn, and against that, most farmers are still spraying with pesticide. Even the farmers that has been planted transgenic crops, they also still spraying with chemical insecticide. So I think it's going to be, if we look at the race and the selection pressure, and also as the 2 previous speakers mentioned, for the device diversity of crops that are available in the field and continuous generation, because we can find corn anywhere, anytime in the country, but we also that this species also can feed on different crops other than corn. So this is just to show you the data that we have been that we published last year about the baseline susceptibility of Cry1A, Cry1A 105, and Cry2AP2 on Asian corn borer, which is good for Asian corn borer, but as we know that this protein is also are intended to controlling the Fall Armyworm. So I guess that in the next few years, it's going to be very busy studies to look at on those two species, Asian corn borer and Asian corn borers, not only to chemical insecticide, or also for the transgenic VT corn. So what about the resistance management in Indonesia, and particularly for VT crops? I think the regulation in Indonesia says that the resistance management is the responsibility of the proponent. And as Dominic said, it's similar to in Indonesia, that every proponent or the technological owner have the right to develop its own resistance management, and they submitted the proposal during the registration process, and then they will be approved by the technical committee. And after commercialization, it's mandatory that each company or each technological owner will submit information in regard with the susceptibility of the targeted pest to the government, to Indonesia, to see whether resistance has been occurring or not. Okay, this is going to be my last slide. So it's a short presentation. Hopefully we'll have a discussion from what we have now and what kind of risk that we are facing right now. What do we need from now? If I think about this, there are several items or several matters or several things that we need. But if I have to choose the top three, I'll put the one that is already on the screen. One is role of the farmers in detecting resistance. And I think by improving, raising the awareness of the farmers, the growers, and also the availability of the simple reporting system will help for not only managing, but also dealing the resistance for particular pests like Fall Armyworm. Because in the past, when I worked with the wheat armyworm, I think I have the benefit for receiving the information from the farmers, and then we can follow up with the necessary action to manage this particular pest. Second one is misreading of the main pest. So we talk about Fall Armyworm and also the Asian corn borer. There is no way that we will be doing or we will be able to do some research on these two species, and we are not having competence. We are not having ability to look after and to do much reading in the lab. And as David says, that the availability of the susceptible population is very important. And luckily enough that we have that population that was collected in 2019. And last but not least is the detection tools. No matter what the detection tools that we will be using, either bioassay, biochemical assay, or molecular assay, I think having the harmonized and standardized protocol, especially for regional and global purposes, including for domestic. So I think it's going to be very important, because using the standardized protocol, I think the data that we'll be sharing to different parts of the world will be meaningful for comparison, and also for taking the benefit from the availability of the data coming from other parts of the world. So I guess that's all I can say, Alison, and I'm happy to respond to any questions, and hopefully we will have plenty of time for the discussion. Thank you very much. And I'll stop sharing.
Excellent. Thank you so much. I'm just inviting someone else to promote to a panelist at the same time. Multitasking, not my best thing to do. So Andy, that was fantastic. Really interesting to see what you had worked on sort of around COVID time and having that Fall Armyworm population there, and then revisiting and looking now. And in between, you've got the introduction of BT as well. I have a question around farmers, because I know that you work or you talk a lot with farmers out in the field. Do you see that they're using less pesticides as they've got more comfortable or more aware of fall armyworm, and it's been quite a few years now, and with the introduction of BT? Are they using less pesticides, and do they rotate their mode of action? I mean, are they aware of that, and are they doing that?
Based on my observation in the field, because I haven't not done yet since 2021 in terms of the with the systematic survey, but farmers are still using 2-3 times for fall armyworm, although they have been planting for with the transgenic corn because they know that the transgenic corn does not protect from fall armyworm. So they still are quite sprays in mainly. And rotation, yes, they do some rotation, but it's very limited in terms of the mode of action that they have. Because in many cases they are more familiar with the branding instead of the active ingredients. But from the regulation perspective, actually the government already required for every pesticide registered in Indonesia to put the classification of the mode of action based on the IRAT classification. So if the grower reads that label properly, and then it's going to be easy for them to do some rotation. But as I guess Dominic and David says, that raising the awareness, education are very important, essential for the grower as well. Thank you, Alison.
Thanks, Andy. I'm just going to throw it back to Ramesh now, and then I've got a couple of other questions I've got coming through as well. And then I'm going to ask one of our colleagues from New South Wales just to talk about one of his responses as well to one of the questions. Ramesh, to you first.
Great. Thanks, Andy, for this presentation. So considering the regulatory and stewardship challenges across different production systems, what guidance would you offer to ensure insecticides are used responsibly without compromising growers' access to essential management tools?
Yeah, I think, as I mentioned earlier, raising the awareness of this problem, I mean, resistance problem in the grower and the ground is going to be very, very important because in many cases, when I talk with the farmers, they may indicate that the insecticide that used to be very effective and it's no longer effective, but it just stopped there. They're just giving the information, but there is no plausible solution or options there available either as a knowledge or as a practical alternative for them. But this kind of information is very important. So that's why for Indonesian perspective, I think it's going to be very important if, simple system is available for the grower to report, because it's going to be very difficult for anyone to collect, so many insects from different population. Even though monitoring is going to be one part, but in addition to regular monitoring, the participation of the grower in the early detection of failure in small plot is going to be very important and very powerful tool for mitigating the resistance in fall armyworm. I think that's what I can respond.
Good.
Thank you for
that's good. Thanks.
Fantastic. I'm just going to throw it. Thanks, Andy. I'm going to throw it over to. I've just invited a new guest into our panel from New South Wales, and there was a question here around: are there reliable molecular diagnostic tools available for monitoring resistance in fall armyworm populations as an alternative to conventional protein-based or crop protection bioassays? And I'm just going to throw it over to, I'm not going to pronounce your name, I'm afraid, Duong Nguyen.
You are right, Duong.
Not close at all, I'm afraid, but that's very kind. You had a good answer. I thought I'd just open it, let you actually tell everyone rather than it's written in the answers there. Could you just share with what you've been doing there?
Yeah, sure. Hi, everyone. My name is Dương Nguyen, and I am a research entomologist. And the name, how to pronounce my name, Dương. So it starts with D,
Zoom.
but yes, that's correct. Thank you. So I am a research scientist, and I'm leading the entomology, insecticide resistance, and genomics here at New South Wales DPI. And I have been working with fall armyworm resistance monitoring since 2020, when fall armyworm first came in Australia. And our team have successfully developed a multi-American sequencing, which is genomic tools which you can use to screen for multiple insecticide groups from group 1 to group 28. And for multiple sample in a single sequencing platform. So that's why it's very cost effective and very high throughput, which I can say. And we published that multi-American sequencing already. And that platform have been used to monitor insecticide resistant in fall armyworm collected from Queensland and WA for several years. So I highly recommended this platform for you in other countries, because we can be able to use that platform for multiple thousands of samples in the same platform. Very effective, very cost effective, and very high throughput. And thank you, Dominic, for your presentations. You mentioned cotton industries have been very successful control monitoring insecticide resistance in cotton. And yes, I can confirm that's correct, because our laboratory have been working with cotton industry for more than 30 years. So DPI RD, so our team to monitor insecticide resistance in cotton via phenotypic, bioassay, and also molecular. Thank you, everyone. If you have any questions, contact me by my email address. Thank you.
Excellent. And her email address is in the chat there. So thank you so much for telling us about that. That's extremely interesting. And it's nice just to pop on and share that right there. I've got a few questions that came in before as well that I thought I would ask as well. And maybe one of you can choose. We'll see who wants to answer this question. But it is, my technical assessment of overseas data indicated only about 70% efficacy of new MOA products. Is this acceptable, and what issues does it cause? E.g., sublethal doses lead quickly to resistance. So is this maybe Dominic? Shall I throw that over to you?
Yes, if you can repeat the question.
Yeah. The question was, my technical assessment of overseas data indicated only about 70% efficacy of new MOA products. Is this acceptable, and what issue does it cause? E.g., sublethal doses lead quickly to resistance. So are the new products that are coming on, are they effective enough?
I guess it depends on what the grower would define as acceptable control. The growers that I work at would not define 70% as acceptable control, but with a pest like fall armyworm in the whirl of maize, it's really challenging to deliver the insecticide to where it needs to be. So I guess it would depend on how effective is it relative to the other products. What's the price point? Can you tank mix it with something else? In terms of resistance management, though, 70% control. My question would be, is that reduced control due to the fact that the insecticide is not that effective? Like there's a proportion that are surviving, that are getting contacted with the insecticide, that are you're actually increasing resistance allele, or is the insecticide just not contacting the insect? So I guess that'd be my question.
Well, thanks, Dominic. And if you are the person that asked that question, I think it might have been Graham Walker from New Zealand. If you're in the room, and you can put your hand up if you want to follow that question up. I have a question for you, Dominic, while you're speaking. You talked about how resistance was found, how you well, you found that there was resistance, but the only action happened when there was field failure. I think that was your sentence. So how do you sort of fix that? I mean, you talked a bit about the behavior and reaching out to farmers, but this is a bit of an issue in that we would be very keen to set up a monitoring for resistance, perhaps at a regional scale and at a country scale, but there's still that issue of even if you discover something, how do you make sure that it gets to farmers and farmers change? They don't need to see field failure to change.
One of the biggest things that I didn't talk about that maybe only I'm a fan of is so I talked about my efforts in extension to change grower behavior, but I think the biggest change is that the U.S. Environmental Protection Agency has also recognized that like the resistance management practices of the past are not working, and they've actually come up with a proposal that will require growers to purchase 20% of their total corn purchases to non-BT hybrids alongside of their BT hybrids. And of course, this would just totally force the issue of resistance management. And then I won't have to worry so much about outreach. But I think it goes to the point of the good Australian management example, right? Resistance management is going to work best when you have the government, the industry who's producing the seeds, and the growers all on board working in tandem. We're certainly not there in the US working in tandem, but there's different efforts in place to increase resistance management. On the company side, they're very interested in genomic monitoring for resistance. So can we get out ahead of this and see if changes are happening over time before we see the field failures? So maybe we'll all get there in different ways.
Excellent, thanks. David, a question for you right here in our Q&A is, do biological insecticides give us effective options for rotation within a resistance management strategy? And if you're on mute at the moment, if you want me to repeat that, it's the last question
Yeah,
there.
you can
Yeah.
repeat the question, sorry.
So do biological insecticides give us effective options for rotation within a resistance management strategy?
I mean, yes, of course, no, but it depends on the threshold or the level or control. But I was a chess player, and I play chess with acceptable level. And then I see all the board, no, I see all the pawns and all the pieces. So I see resistance management like it's not the deploying of the queen, it's all the measures, no? All the factors, but all of this is in economical terms, no? How expensive is to use an alternative tool, and how I mean, is in some cases, in case of corn, the margin of benefit for farmers, at least in the Latin America, is very thin. And I don't know how it's in Asia, the profits, but it's very razor thin, the profits. And that's the problem. If it's costly, then it's not possible to get profits for the tools that are used against Fall Armyworm.
Excellent point. David, I like the chessboard analogy. Having all the chess pieces maybe on the board is important. The tools in the IPM toolbox. Dominic, I really liked you've actually had a really nice ending there around how everyone has to work together and the different players. So it's a very sort of, you guys are drawing this session to a close nicely. Before I hand over to Ramesh for sort of a closing thoughts, I just want to ask Tech, you've been listening, and I know I just heard genomics kind of raised there around for resistance, etc. Any thoughts from you on the session?
Yeah, a little bit, I guess. It's been a fantastic presentation. Again, thank you to all the presenters. In terms of the scenario in Australia versus Brazil and US, ultimately there's a fundamental difference in that Australia is a small country, even for the population size, especially so for the GM cotton. So managing the smaller grower groups that has an interest in the BT production, the BT management approach and reducing resistance is, I would say, easier. And legislation set out is that the farmers can see that they need to come together. This differs significantly across, say, Brazil and the US, and even so for Africa, Southeast Asia, and whatnot, because now you're talking about many different countries across a connected landscape. So getting all the different countries to agree to a specific type of cultivating regime, especially concerning BT versus non-BT crops, will be significantly challenged. On top of that, we actually don't really understand in the new landscape, especially the introduced landscape, how the fall armyworm populations are connected in terms of gene flow. So does a population that has developed resistance would likely to spread the resistance allele to the population elsewhere? So we don't really know a lot about it. There's a lot of conjecture about long distance migration and whatnot. Again, those need to be backed up by genomic studies, which we are not there yet, and climate factors. So again, it's quite different in Southeast Asia versus, say, US. So yeah, look, this is why we're here. We're here to work together to come out to see if we can, further push our knowledge further to see how we can actually come up with a sensible management solution for BT, for example. Thank you.
Excellent. Thanks, Tech. Andy, you've got your hand up. Is that because you wanted to say something?
Yes. I just want to add something in regard with the biopesticide. I think talking about the biopesticide, about the potential, I think they are very good in terms of specification and probably the efficacy under the probably laboratory or more controlled environment. But in many cases, at least what I've been seeing in the field, efficacy is something that is in questions because the application of biopesticide probably require more precision than synthetic chemical pesticide. So the potential, yes, but in reality, sometimes it's more challenging than using chemical pesticide. But improving the precision in application will be very helpful for the effectiveness of this material, I guess. Thank you.
Excellent. Thanks, Andy, and thanks for adding that in. And all really good points. Thank you all. I'm just going to say quickly, there's quite a few questions on bioassays as well, and I know Andy's a bit of an expert on that and has actually given a masterclass session earlier this year. So I will be sharing those resources with you, and Ramesh and I will also follow up with some of the other questions that we haven't got around to answering today. I'm going to throw it over to Ramesh now just to give us some closing thoughts quite quickly. We've got 2 minutes, but we might just go over like 3 minutes over. Just so he's not too stressed.
Thanks, Alison. Thank you all for your engagement throughout today's session with almost 249 participants from across the globe. The presentation and discussions have highlighted both complexity of fall armyworm resistance and the value of coordinated evidence-based approaches across regions to manage fall armyworm resistance in area-wide management perspective. As an extension officer working closely with growers, I find it encouraging to see research insights, field experience, and regulatory perspectives brought together in such a practical way. We appreciate the contributions from our speakers, Dr. David, Dr. Dominic, and Professor Andy, and accepting our proposal to come to this webinar. Your questions and reflections from the audience have also added important context for how this knowledge can be applied on the ground. Thanks to Dr. Tech supporting in Q&A and your thoughts. Thank you once again for your time and participation. We look forward to continued collaboration with Alison and all of you as we work to strengthen fall armyworm management across our industries and regions. I will now hand back to Alison. Thank you.
Yes, thank you so much, Ramesh. Thank you for summarizing and giving that sort of end statement. And I would just also like to say thank you very much to all our speakers. Wonderful to have you, and very big thanks to everyone who participated today. It's huge turnout for just before the end of the year. So as we close 2025, I just want to wish everyone good luck and looking forward to seeing you in the new year as well. There are some questions that are not answered, and that's okay because we will take the time to make a selection of those and provide some responses and share those responses back with you. So there's still some very interesting questions there that we will come back to you on. So thank you very much. Have a very nice afternoon, day, evening, sleep for our US colleagues who are at the end of their day. Take care, and we'll see you next time. Thank you.
Thank you.
Bye.
Thank
Thank
you so
you.
much.
Bye.
Thank you.
Bye.
Bye-bye.
Bye.
Bye.
Bye, everyone.